Even though this is our inaugural conference, a lot of work has been done already, perhaps during the conference by a team of us ever since we started data science about 3-4 years ago. And you'll hear about some of the activities we've tried to develop to make data science a good place on campus. You'll hear about our research centers. You'll hear about our educational efforts. You'll hear about how we're trying to increase multidisciplinary aspects of research through graduate fellowships and postdoc fellowships. You'll hear about outreach activities. But I think the past is not what's most important. What's most important is the future. And we're very excited to have you all here because we just want to learn from you. We want to know how we want your help in making Stanford data science better. We want your help in energy and enthusiasm in telling us about the work that we need to do. And Stanford data science cannot happen without you. And so we're very happy to have this interdisciplinary conversation today so that we can learn together how we can fulfill your expectations and be a positive place on campus, a place that values people doing data science. So I'm expecting a lot of things from today. We've organized this day as a conversation about data science. We want to increase our sense of community and shared intellectual goals. Now, we'll hear a lot about many different topics, and we'll start with neuroscience. And it's my great pleasure to welcome Emery Brown to Stanford. Emery is many things at once. He's an anesthesiologist, he's a statistician, and he's a neuroscientist. And he's a Zappel Professor of Anesthesia at Harvard University and Massachusetts General Hospital. He's a Professor of Computational Neuroscience at MIT. And he has something very unusual on his resume. He's a member of all 3 academies. And I don't know of how many people I can say this. He's a member of the National Academy of Sciences. He's a member of the National Academy of Engineering. And he's a member of the National Academy of Medicine. I think when we thought about this conference, we could not think about a more exciting speaker because Emery's work is about consciousness. And what is consciousness? It has to be, in my view, one of life's greatest mysteries. And what does it mean to be aware or to sleep or to be in a state of deep coma? And so this is broadly speaking, Emery's field of research. And I think Emery has sort of brought the issue of consciousness out of science fiction or even poetry to the realm of science. And so he's been interested in the brain and all states of consciousness. As you'll see perhaps from these lectures, there's a continuum of states in consciousness. You can be unconscious and not perspective. You can be unconscious but perspective. And in his research, he has used data in this particular case, EEG, that is electroencephalograms, to infer the level of unconsciousness in an individual, for example, under general anesthesia. And so he has shown after a remarkable study in 2013, how anesthesia hijacks the brain waves and makes it difficult for different parts of the brain to communicate, and that's what leads to unconsciousness. So his work on anesthesia brings us a window on the brain, and we can learn about the brain, again, perhaps one of the biggest mysteries, how the brain is working by understanding and monitoring data as we put people and patients through induced unconsciousness. Emery, it's a pleasure to have you here. You're a great friend to Stanford, and we look forward to your lecture.
Great. Well, thank you so much. Let me just get this. So, thanks so much for having me. It's a real pleasure to be here, and I hope I don't make you unconscious during my talk from what I'm about to say. So, I'm going to focus. I will get to some issues of consciousness towards the end of the lecture. But what I want to talk about is this sort of general theme of keeping science and data science. And to do that, I'm going to talk about 3 topics. A little closer.
Here's your notes. I need that full screen. Thank you.
Oh.
Just run like that.
Oh, okay. All right. That was an audible. All right. So I'm going to talk about measuring the period of the human biological clock. I'll say a little bit about what it's like to decode information from the rat hippocampus, and then I'll talk about monitoring depth of anesthesia, where we'll discuss some issues of consciousness. All right. So measuring the period of the human biological clock. So I was a graduate student. I did my PhD in statistics at Harvard, and one of my PhD advisors was Peter Huber. And as many of you know, Peter was and still is an expert in robust statistics. So I went to Professor Huber when he got there, and I said, Professor Huber, I'm interested in doing a PhD in robust statistics. He goes, I'm tired of robust statistics. I said, I'm tired of robust statistics too. And he said, he said multiple time series analysis. He said that's an area where there's a lot of things to be done. And I said, oh wow. So I said, if I find a problem, multiple time series analysis, will you work with me? He goes, well, if you find an interesting problem, I may work with you. If it was not so interesting, I may not work with you. So I said, okay, fine. So I was very lucky because Chuck Seisler had just come from Stanford, actually, and set up his laboratory, his sleep research laboratory, at the Brigham and Women's Hospital. And Chuck was studying the human circadian system. And he had these ideas for these really, really cool experiments. So I'll just show you some of the data. He was going to be doing 3 types of protocols. So one was the constant routine, where people were sort of in a room in a same position, sort of no meals or that sort of thing, or at a very sort of stable condition, kept awake for about 30 hours or so. Because what he wanted to do was track their circadian rhythm, following their body temperature, and then really trying to estimate where the minimum occurred, because that was the main phase. That was the phase in the cycle which was most robust, let me just say it that way. Then he was thinking about sort of some of the more standard, longer protocols like free run, where you isolate someone in an environment, they have no light cues or this, in other words, they have no cues from the outside world. Their intrinsic clock runs its intrinsic period, and you do some analysis to measure the period. And then he was thinking about a forced desynchrony protocol. With forced desynchrony, you put someone on a daylight cycle where they can entrain, so put them on a 28-hour day. All right? So on a 28-hour day, your internal clock can't entrain to 28 hours, so you'll actually get to see the actual intrinsic period. And you can actually, and so these are these sorts of protocols he's going to be doing. And he's recording many things, but the main thing is human core temperature, measured from, just to be frank, from a rectal probe every minute. So these were very interesting experiments. All right? So this was the first paper that I wrote of that work. So you can see, because of the data, that everything's harmonic, so no surprise I'm working on harmonic regression, all right? Not really sort of straightforward, obvious model. And you see the waveforms are asymmetric, so working on harmonic regression models that have more than one harmonic, at least 2 harmonics, let's say, all right? So just to make the point clear, go back to what I call Statistics 101. So I've got some data. I know the mean, I know the variance. I take the sample mean as the estimate of the true mean, and the variance goes down as 1 over n. Okay, so why is he telling me this? This is Statistics 101. Okay, all right. Because of this. So it turns out that if you have a harmonic regression model like the one I'm showing here now, and so it's a harmonic regression, we have at least, we have p harmonics, and we have, let's say, just a simple correlated noise model, all right? Then if you calculate the variance of the period estimate, it goes down as 1 over t cubed and not 1 over n, or 1 over t, all right? And this was known at the time, this was, been known for years. Peter Bloomfield, in his book on time series analysis, talks about this and gives some intuition about why it's the case. And the quick back-of-the-envelope intuition is that if you compute the Fisher information for this problem, right, and you look at the sum, you get a sum of t squared. And the sum of t squared is proportional to big T, cubed, so that's where it comes. It's really just that simple. It's not deep at all, all right? But what it means is if you do have a model where this model works, you can get very precise estimates of the period. That's the key thing, all right? So, and if you look right here, you see this? You have this thing which is like a signal-to-noise ratio. Here's the energy at the harmonics, and here's the spectrum at that same location, all right? Well, so this is the first paper I wrote. It was a note to Biometrica, and I sent it off in September. It was accepted in December. I said, man, this academic thing's going to be a piece of cake. Yeah, I got this, all right? And so I just checked, literally just before I started here, and as of this morning, it has a whole 21 citations. All right? So it's rocking the world, as you can see. All right. So that was in the early 90s and late 80s. And Chuck decided around the mid-90s that what he wanted to do was measure the period of human biological clock. He had a really cool biological intuition. He realized that the period was being improperly estimated. And so here's the logic of what he thought. All other species had periods that were closer to 24 hours rather than 25, all right? And he just couldn't understand why this would be, that humans would have one closer to 25. And then he realized something important. We'd published a paper back in like 1989 looking at the effects of light on the human pacemaker. And he basically showed that light was one of the most important factors for shifting the internal clock, all right? It could be artificial light, it could be natural light. And so during these free-run studies where you have someone in isolation, you select your own light-dark cycle. So what that means is that you're giving yourself light as an input to your circadian oscillator, and there's an asymmetry in the way light affects the oscillator. And the way it happens is it tends to delay the oscillator a little bit as opposed to advancing it. So what happens is that when you do these studies like this, you get an incorrect estimate of the period. It's a little bit longer than it should be. So that was Chuck's insight, a really, really cool insight that he had with Richard Cronauer. All right. And so these are those microphase shifts. And so I told him that, well, you know, if you do this, you're going to have some way to actually measure the error because you're saying it's this, but it's plus or minus something, all right? So these are some of the so he did this in forced desynchrony. So here's what the forced desynchrony protocol looks like. So the person is on a 28-hour day, all right? Now they're 14 hours of 10 lux of light and then 14 hours of total darkness. And if you ever, 10 lux of light is basically practically darkness in and of itself. And the other thing is, but we probably have more light in our room now just from having our cell phones on, our radios, our televisions on, or whatever. But this is really, really, really dark. All right, so he's really kind of taking out the effect of light on the pacemaker. And you can entrain to, or a human can entrain to a period of 28 hours. So you're going to see the intrinsic period of the oscillator. And you can actually see that in the data. So these are 5 subjects here from that study. And if you look here, you can see the constructive and destructive interference. There are 2 big components, a 28-hour component and something around 24-hour component in this data, in each of the subjects. You can see it there. It's like, for example, like right here.
We cannot see your cursor.
Right. That's why I switched the other mode. Okay. That's why I switched to the other display mode. All right. But so look at subject A, between 15 and 20 hours, you can see that it adds up destructively there. Whereas if you look at the same subject around 15, they're enhanced. So it's getting bigger in one place and small in the other. So that's the constructive and destructive interference of these 2 oscillations. There are 2 oscillations there. That's the bottom line. All right? So here's the model. Now we just add a 28-hour component to the model. There's the mean. That's the circadian piece. We have at least 2 harmonics for the circadian piece and about 4 harmonics for the 28-hour day oscillator. And we used correlated noise because it's very real. For example, your body temperature is highly correlated from time to time. And taking that correlation into account makes the estimate of the period much more accurate because you don't have like 36,000 independent data points. They're highly correlated. You have to take that into account, all right? All right. So here's what the decomposition looks like. This is for one subject. You can see the circadian piece, and the magnitude of the 20-hour component is as big as the circadian piece. And when you add them together, now you can see the constructive and destructive interference really clearly as they're cleaned up there a bit. And then the residuals together with the AR processes in the bottom panel. And I can tell you we were able to convert each one of these subjects' data into white noise with the model. In other words, the model fit very well for all the subjects. So we published this paper in Science back in 1999. And so I was able to dust off my error formula. You know, it's been cited 21 times. You know, and we had a very precise estimate for the period. That was kind of the take-home message. And it had this really, like I said, this really cool feature, the signal-to-noise ratio piece in there. And so here's the data from the subjects that we looked at. And actually, Chuck studied about 12 sort of normal, healthy, you know, sort of young people between the age of 18 and 64. And he studied, believe it or not, he studied 12 elderly patients. We had 12 patients, 12 subjects who were over the age of 65 that underwent this protocol, believe it or not. Because we thought that the question was, were the periods of older people different from those of younger people? That was the issue. And you can see that those are the period estimates for each of the subjects, and the standard errors are not in hours, they're in minutes. And it's because of the protocol. The model worked and gave you a very accurate description of the data. All right? And then Chuck had one subject who did both protocols. The subject did a forced desynchrony protocol, okay, and he did the free run protocol. And so I told him that if you had the light times from the free run protocol, you could actually estimate the period from that data as well. And so that's what I did. So here's the model that I worked out. It's a Van der Pol oscillator with light inputs. So this is the light input going into the Van der Pol oscillator. So you have a signal-to-noise model here where the signal is the solution of this Van der Pol differential equation, basically. Just like the other situation before, except now the signal is represented by the Van der Pol, and it can take light as an input. All right, so when you do that, so so let's look at this one over here. Here's the harmonic regression. When you ignore the light effects, you get these huge oscillations. It looks like the circadian oscillation is really big. You know, there's the noise. But when you take into account the light, here are the light effects right there. And then here's the circadian oscillator, has a much smaller amplitude. And look at the period. The period is 24.25 hours. So you could actually extract it from these data with the proper model, essentially. And over here, if you did it the naive way, you get the mistaken estimate that it's 25 hours instead of 24. That's the bottom line, all right? So I was feeling good about this until last year, all right? And this paper came out in Scientific Reports. And it said they had a data-driven, unsupervised time series modeling approach so you could estimate circadian rhythms and their properties from skin surface temperature in free living conditions. Promises everything. And not only that, they said they could divide people into 3 distinct clusters. All right, and so this provides, as they said, a novel basis for modeling and understanding the human circadian function in free living conditions. Now, just what were the limitations of their study? So the first one I put up there, they only used a single harmonic. And you saw that in the data that I showed you, you had to have at least 2 harmonics to describe the circadian pacemaker, and you had to have another piece there to actually, if you had the 28-hour day component. These other 3 points, they're their own. This is what they said in their paper about their limitations, not me. Look at the first one. It lacks measurements of light exposure, which would be a very valuable variable to include. I think so, right? The crucial question on how these various factors that they listed just above that, you know, interact in circadian biology cannot be evaluated from the study design. And so I like the last one, though. It says, the concept of circadian period thus refers here to a broader concept of periodicity than that assessed in the controlled laboratory study. Broader concept? I mean, I think about how hard we worked to, you know, Chuck doing those studies, recruiting all the subjects, you know, curating the data and making sure the data were properly collected and just obsessing about everything to get the results that I just showed you. But now, we can do it with just skin temperature. Don't have to use any of the complicated models like we used, and it's a novel approach, and I'm sure it's going to have huge impact. And I actually looked at the article to see if Chuck's name appeared in there at all, Seisler, and it appeared once in a paper where he was like the 15th author or something like that. So none of his major work was actually looked at. And I just ask you the question, is this data science? All right? So let's move on to another topic, decoding the information. I guess the answer was no, right? Okay, all right. So decoding information from the information stored in a rat's short-term memory. So I'd worked on circadian rhythms for a number of years, and I decided to make a career shift. I decided to find another area to become involved in, and I thought about it fairly carefully about what areas I wanted to attack. I thought about genetics, I thought about the environment, women's health. I thought a little bit about maybe endocrinology in particular. And then I thought about brain science, and I thought I would get involved in neuroscience. And so I had to retrain. I didn't know much about neuroscience at all. I'd been to medical school and that sort of thing. So I took the summer course at Woods Hole in the marine biological laboratory. The Methods in Computational Neuroscience course was run by David Tank, who was at Bell Labs at the time, and David Kleinfeld, who was there then as well. And I just had to learn enough. I had to learn more. I didn't know enough to really enter this field. And then David Kleinfeld, we had to do a summer project. He said, why don't you work on the problem of decoding information in the rat hippocampus? I didn't even know what he was talking about, but I thought, okay, I'll study that. And it just so happens that one of the young experts in the field at the time had just moved to Boston, and that was Matt Wilson, right? He's now at MIT. And so the cool thing about the hippocampus, that's not where the hippopotamus go to school, all right? It's a seahorse. It stands for seahorse or sea monster, because if you look at it, you can see where they've taken off the part of the rat brain there, and it looks kind of like a seahorse or a sea monster, all right? And so these neurons, actually, before I tell you that, there's another actor in this story who was Lauren Frank. And again, this speaks to data science. So at the time, Matt was on my list of people to talk to in Boston. And I didn't know enough to really talk to him yet. So I was teaching this course with Art Dempster at Harvard on statistics and physiology. And we devoted the second semester specifically to neuroscience as a way to learn neuroscience, for me anyway. And so I was learning things one lecture ahead of teaching it to the students. And so one day, we get this call from MIT, and it says, some folks from MIT want to come to your class. And I said, sure, that's perfect, that's fine, that'd be great. And it was Lauren Frank and Ken Blum, who were in Matt Wilson's lab at the time. And the reason they were coming was because MIT had no statistics department. And so they were looking for help with their hippocampal data. I was like, wow, this is perfect. So that's actually how I got involved with working with them on the hippocampus. And one of the things about the rat hippocampus is and the hippocampus in general, it's the area of short-term memory. So, and the rat hippocampal neurons have this really cool property called place cells. That is, as a rat runs around an environment, yeah, that might be a good idea. Thank you. Well, I'll just work out here. But now the mic's here. I'll just stay put. All right. So the rat hippocampus has this really cool property. That is, as an animal runs around an environment, neurons start to fire in very specific places in the environment. And they're called place cells or place receptive fields. And I can just show you that here. Let me play this little video here. So you can see there's a red area forming. You're going to see a white area forming. And the red area forming is where one neuron, every time the animal is in that part of the environment, the neuron is spiking. And then there's another neuron, the green neuron, which every time it's in the lower part of the environment there, it's spiking. And there's a white area. So you can actually see there are three areas defined. These are place fields. So the red neuron, it says, I'm kind of up here somewhere. The white neuron says, well, I'm kind of down here. The green neuron says, I'm over here. So it's a very simple question you could ask. if you had enough of these neurons, could you actually read where the animal was in its environment just from the spiking activity? Could you basically decode the information that it's showing you, all right? And so does the animal maintain a dynamic representation of its position in the ensemble spiking activity in the hippocampus? So here's what the experiment looks like. So what you do in the first part of the experiment, the encoding analysis, let the animal run around, just like it was doing. Runs around, runs around, runs around. And you record the spiking activity, all right? And then from that, you do that, let's say, for about 15 minutes. And then from that part of the data, you estimate the place fields, where are they located, all right? Then you take the last part of the data, the last 10 minutes, and now, using the place field estimates, as the animal runs around, you estimate where it is in the environment just from the spiking activity. All right, so that's what we did. So we had about 30 neurons, and this was a big deal at the time, having simultaneous recording from 30 neurons. That was really huge. And so these are some of the results, just the pictures of the results that Matt had published in 1993 in Science, showing that this was feasible. So that's an animal running around. The black line is where the animal actually is, and the red line is the estimate, all right? So we did some work on developing a slightly better model. And again, this goes to, like, I was learning this stuff. I mean, I hadn't worked on point processes when I was a PhD student. It was something I learned just for this particular problem. So representing the ones and zeros, the spikes, as point processes is what we took on. And we needed some sort of state-space model. So we just went back to the Chapman-Kolmogorov setup and Bayes' theorem for basically what we now call state-space modeling. And using Bayes' rule, I want to get the best estimate of the animal's position given all the spiking activity. That sort of makes sense. And then what I'll have to do is I need some sort of one-step prediction. So I guess where he's going to be in the next time period. And then that becomes my prior, and here's my likelihood. And so initially we used an inhomogeneous Poisson likelihood, where we model the shapes of the fields either as Gaussian shapes or we use what are called Zernike polynomials. Zernike polynomials are orthogonal polynomials on the disk. And they're just like, Fourier series on the line. These are orthogonal polynomials. Not so much just because we're trying to get the shape of the field right, all right? So you work through that, and you end up with something that looks like a Kalman filter algorithm for point processes, basically. It takes as its inputs the spiking activity. It measures the difference between the spiking activity that's predicted and the spiking activity that you actually see. That's an innovation. You add that to the previous estimate, and then you get an update. And now while this looks linear, it's not linear because xk is also in lambda c on the right-hand side of the equation. So it's a little nonlinear equation to solve, but it's basically this. I wrote it this way. You can see that it has the same formula like a Kalman filter, essentially. All right? So here's the estimates. Here's the analysis of the fields. So there are two animals, animal 1 and animal 2. Animal 1 is column 1. Animal 2 is column 2. And then the Zernike polynomial fits. So there are 34 cells for animal 1, 32 cells for animal 2. And you see the Gaussian fit and the corresponding Zernike fit. They're similar. And in the places like for the Gaussian fits where you see sort of two ovals, what we did there is we just fit two Gaussians because you could see there were two clumps of data, all right, that you had to account for. All right, so now these tell me for that neuron the probability of spiking given the animal's position. So now I've got this over an ensemble of 30 neurons. Can I now invert this with Bayes' rule and just estimate where the animal is? All right. So let's see. All right. So here we are. All right. So I'm going to show you the rat running around first. That's about the size of his head. The environment is 60 centimeters across. All right. And actually, let me just do it this way. Sorry. I just want to show you the rat first. Okay. Here we go. He looks like he's on cocaine, basically. Because these newer machines, this is nothing for them to run through this. But the green activity on the right is the spiking activity. That's where, they threw little food pellets to make him run around like that so you can, Cocoa Puffs actually works best, it turns out. All right. So, but that's the experiment, all right? And that's basically 10 minutes of data, all right? So let's start it back. So now let's look at the Gaussian estimate. So when I say Gaussian estimate, I mean I built the fields based on assuming a Gaussian shape for every field, all right? So here's that. And down below here, what you're going to see is the coverage probably. See, the animal's running away from the estimator, or there's lag, right? All right, but you can see that it's right in the area where he is, but it's not sort of right up on him, okay? And below, you see the red line down at the bottom where it says 39. That's the instantaneous coverage probability. If it's working perfectly, it would be 95%, all right? So it's not quite 95%. In fact, it's a good distance away from it, but it's not doing too bad, all right? So let's go back here. And now let's look at the Zernike polynomial one. So just the different shapes of the fields. And the coverage probability will be up top again. So that you can see both of them now. So the Zernike intersects much more frequently with the animal itself than the Gaussian one does. And the coverage probability, while it's not 95%, it's 70%. So he's doing better, certainly doing better. But what's interesting about it, this is only from 30 neurons. And this is a very naive kind of, no-frills statistical model. It shows you there's a tremendous amount of information in these neurons there, so that if you really take the modeling of it seriously, you can probably extract even a lot more. All right, so I'll stop there. So here's a summary. So the error for that time period for the two animals was around 7 or about almost 8 centimeters for the Gaussian model, about 6 and 5.5 for the Zernike model. The reverse correlation technique, which is just basically, it's an ad hoc procedure. I'll skip on telling you how that's actually done. But that's what Matt used in his analyses. He got an error of about 30 centimeters. And even in his paper that he showed, in the ones in Science, the error was about 30 centimeters for a very similar environment. So this was a tremendous improvement just by doing a little bit of statistical modeling, just to get the shapes of the fields right and something about the spiking activity, better described. And so we feel pretty comfortable to say that, yes, the animal does maintain this dynamic representation. And then I felt like, there's one thing I forgot to tell you. So one thing Peter Huber told me, he said, find an interesting problem and try to squeeze all the methodology out of it you possibly can, all right? And I felt like that's what happened a little bit here with this research. We were able to develop this point process sort of Statespace model framework and then use it not only here but on a lot of other problems. I just listed a few of them here. So I felt that this is the sort of thing that we as, in this case, statistician or data scientists, should be doing. What I found is if I take real problems seriously, then first it tells me what the assumptions I need to make are. They're much more interesting than ones I make mathematically. And then also, as a consequence, I usually find broad applicability, all right? So let's swing to monitoring depth of anesthesia, all right? So, and I put this up here like this on purpose, because this is how people talk. When they say things like depth of anesthesia, all right? So what is depth of anesthesia, all right? Okay, so and this is me trying to describe the way people talk about, and people, I mean anesthesiologists, right? Talk about depth of anesthesia. It's an inference being made by an anesthesia caregiver regarding how well a patient is likely to tolerate surgery. It's based on physiological measurements, heart rate, blood pressure, oxygen saturation, movement, the anesthesia drugs the patient has received, the anesthesia caregiver's practice, habits, and experience. That's depth of anesthesia, okay? There's another answer. A number between 0 and 100, computed from an electroencephalogram using the bispectral index, BIS. 100 means you're wide awake, 0 means you're in a profound coma, 40 to 60 is where you have to keep them. Isn't anesthesia easy? All right? So this is developed by the company Aspect Medical back in the early 90s. And that's how it's marketed, even today. That's what you do. You keep someone between 40 and 60, they're well anesthetized. That's all you need to know, all right? So I thought it might be worthwhile kind of dig back into how they could have figured this out. So how does BIS work? No one knows. Because the algorithm is proprietary. It's a black box, all right? And in fact, one of the things that a lot of literature has been wasted on over the last 30 years or so since this came out is trying to guess what's in BIS, all right? So I went to the current manual, the company now is owned by Medtronic. And here's what they said. So they have the EEG, which they take. They compute a power spectrum. They compute a bispectrum. And they compute two things, what's called new suppression, and they compute suppression measurement, also just a suppression measurement. They put that together somehow, and they give you a number like 40 that they're showing there, all right? So now, the spectrum makes sense because there's some frequency content. There's very strong frequency content in the oscillations, all right? Bispectrum may or may not make sense, but if it does, it's because you're measuring, are the frequencies coupled, right? Is there coupling between them? And you might think, well, as the system starts to organize because, you have these oscillations at very fixed frequencies, that things start to couple and move together, all right? Reasonable. The suppression piece is an empirical thing. You'd have to know what EEG data looks like. But when you go into a very profound state of coma, this is one of the signatures that you see, a burst and a suppression, a burst and a suppression. It's a very profound state of coma. It can happen with hypothermia, head injury, anesthesia. And so you're saying, well, if you're in that state, you're really deep. So that would be good to know. And then if you're completely flat, completely suppressed. So that's what the manual right now tells you is in there, okay? So one of the things that it puts emphasis on is the high frequency content in the spectrum, right? It looks at that it puts a lot of weight on that. And so these guys in New Zealand back in 2004 took data from patients who were under anesthesia and measured their bispectra, and they basically found that there was really nothing beyond what was in the power spectrum that was in the signal, essentially. If you had the spectrum, you pretty much had the whole thing, all right? All right. But BIS is predicated on using the bispectrum. They put a lot of emphasis on the fact there's coupling between the frequencies, during this. That's how they market their device. And they modified the upper frequency range, at least in the most recent issue of the manual, to say they look at frequencies between 14 to 30 hertz as the highest frequency, and they also look at the amount of low frequency synchronization. All right. I'm showing you, actually, I have to tell you one other thing. Hold on one second. All right. How is BIS validated? And that's interesting to, if this thing can produce this information, which is so important, how is it validated? This is one of 4 studies, all right? They had 38 patients, and they're randomized to getting 1 of 3 different doses of propofol. Propofol is one of our most used anesthetics. Rendered you profoundly unconscious. It's the anesthetic is unfortunately, responsible for Michael Jackson's demise, all right? And so now what they do is they have 6 leads of EEG, and now here's the stimulus. These are people who are coming for surgery. Nobody would volunteer for this, okay? They get a 2-centimeter skin incision, all right, to start the surgery. And it's at a very precise time. And they measure movement, all right? So the stimulus is getting a skin incision. Did the patient move? Then they look to see what did the EEG do around that time. So that kind of straightforward, nothing deep, all right? And then they looked at BIS. Oh, and that's the thing. They don't tell you how they calculate the outcome. They said they just measured the BIS. They looked at the delta power, so the power between 1 to 4 hertz. The power above, the frequency that divided the spectrum into the 95th percentile, and then the median frequency. So they had these 4 measures that they were computing. And I just wanted to put this little detail in there. So what they did was, so you have 2 groups. You have the people who moved and the people who didn't move, all right? Now you make some EEG measure, and your EEG measure in their case is the BIS. So they put that in a linear discriminant analysis, okay? A linear discriminant analysis. Then, after calculating the linear discriminant analysis, they plugged that into this formula. They did a logistic transform on it. They didn't do a logistic GLM. They did a regression and then plugged this in there. So that's why the numbers scale between 0 and 100, all right? I see you're believing a lot in this right now, all right? There was no separate validation set, and there's no separate test set. In fact, what they did was they had 195 patients that they had studied before. They added these 38 into them. They redid the analysis. So the predictions, the people that the model was fit to are the same people the model was being predicted on, all right? And here's their key slide. Look at the one up top. So this is their key result. See how the bispectral folks, the movers had basically higher BIS, right? They're more awake. And then the patients who didn't move had lower BIS. And then their area under the curve, their AUC, if you look down in the lower panel, is 0.81. Okay, so those are the results. So they had about 4 studies that were very much like this. This is the validation, all right? I'll just be frank with you. I think this research has set our field back 30 years, all right? And one of the big reasons is the first point here. It's a black box. No one knows what's being computed. So you either use it blindly or you ignore it or you're skeptical about using it, all right? And it's been shown that it can be misled by high-frequency noise. So for example, and this is why we have human studies committees, but there's a certain country, I won't name it, where they had medical students take muscle relaxants while being awake. And the muscle relaxants, when their muscles relaxed and they put the BIS on, the BIS went down. They're conscious but paralyzed, all right? Obviously, this is totally unethical. But this is to illustrate the fact that this machine actually reads change in muscle artifact and has nothing to do with EEG associated with someone's level of consciousness, all right? And then, this is interesting. So in a 2004 paper in Lancet, BIS was considered to be very effective against preventing awareness. In a 2008 paper, it was shown that it wasn't. All right, so it's not clear what it supposedly does. And then for 2 drugs, ketamine and dexmedetomidine, it doesn't work. It doesn't work in kids. And the other thing is it has a delay. Okay, so the person's waking up now, right now. So I just waited about 12 or 15 seconds, right? The delay here is sometimes 30 up to 2 minutes before the effect actually shows up on the monitor, all right? And then we estimated like what the use of, so the other thing is anything associated with the EEG, even the work that I do, people say, in fact, I was in a meeting the other day with one of my colleagues, one of my neurosurgical colleagues. He says, that BIS monitor you guys use. I'm like, dude, we're not using the BIS monitor. We have an EEG monitor. All EEG monitors aren't BIS. So anything associated with the EEG in the operating room is called BIS now, all right? So you can imagine what their opinion of it is. And we've estimated that even at our hospital where we have proper instruction on using the EEG, we have somewhere between 23 to 30% of the anesthesiologists actually use some form of EEG, at least the way that we're trying to teach it, all right? And all this starts because of 2 things. And I've presented this to you without any diagrams because that's how this is presented. Nobody's looking at data, all right? If you look at data here, just do some simple plots like I'm going to show you. These questions clean up pretty quickly. But I want to show you this conceptual issue. So what is general anesthesia? First of all, depth of anesthesia doesn't make sense. Anesthesia has like 4 components. You need to be unconscious. You need not to feel pain. You have to have amnesia. You shouldn't move around. And it has to be reversible, and you have physiologic stability. So if you think about what they're doing in these studies, they are putting in a painful stimulus and using a painful stimulus to infer whether or not you're conscious. And your cognitive neuroscience colleagues will tell you if you want to check consciousness, you have to do a cognitive task. Are you consciously processing something or aren't you? So the task, in other words, the definition is totally screwed up. So, and the other thing is, what they're really measuring is some measure of pain, what we call antinociception. It's called antinociception when you're unconscious and pain is still being processed through the body. That's called antinociception as opposed to analgesia. It's a technical difference, but essentially it's a test of pain, right? They're sticking you with a probe, all right? Looking at some data cleans this up in 2 seconds, all right? Here's the deal. This is someone getting propofol, going through different stages of anesthesia. Without even going into details here, when you're awake, you have high frequency, low amplitude oscillations, all right? As you get down to these states here, which are states of coma, and you're unconscious, you have very large amplitude, low frequency oscillations. So anything with any kind of measure which would just track that is going to probably get you 60 or 70% of the way there to saying someone's unconscious, not deep, all right? And you can see the bottom plots down here? These are the two that the BIS includes explicitly. They say they include explicitly, and you can see why, because if these are present, it says you're profoundly unconscious. All right, now, this is my favorite, ketamine. Ketamine really, really messes with the BIS. Really, this is totally cool, all right? So high-frequency oscillation, you're awake, all right? All right, so ketamine says, I'm going to mess with you. I'm going to be profoundly unconscious. I have a high-frequency oscillation and then a slow oscillation, a high-frequency oscillation and a slow oscillation. It goes back and forth between that. Actually, it's really beautiful. If you look here, see the high-frequency oscillation? There's actually a downward chirp. It goes down, goes to low frequency, then jumps back up, and does that. So if you give someone ketamine so that they're profoundly unconscious so you can operate on them, this is what the EEG does. So the BIS can't track this. It gets confused. In fact, it usually reads high. It usually says the person's awake, all right, when you know they're profoundly unconscious. And here's the other drug that it fools too. It's called dexmedetomidine. So the key part is right down here. At high doses of dexmedetomidine, you get the large slow oscillations, right? But if you shake this person like this, they'll wake up. So they're just sedated, all right? So every anesthetic, but remember, they said it doesn't matter what drug you use. If you get a number between 40 and 60, you're unconscious. And what they never thought about, they never looked at the data really, and never thought about the neurophysiology of the drugs. And this is the final one. With kids, it doesn't work in kids. So it makes sense. If you build a model or an algorithm using like the 30-year-old and the 50-year-old and the 80-year-old, the patterns are completely different from what you see for the kids. But the thing is, you can just see this. I mean, this should never have happened to begin with, all right? It's the same spectral structure. There's a lower band, there's an upper band, but the region over which where they lie changes the function of it. It changes very, very systematically. It's not random at all. So this is all knowable, but what you can see is they never plotted any data. All right. So what's my summary here? So each of these anesthetic classes has a different EEG signature. They change systematically with dose, patient age, and physiological condition. So we recommend that everybody use the EEG to take care of patients, but guided by not this sort of rule of thumb, but using the neuroscience behind how the oscillations are generated and what their effects of the drugs are in the brain. And you can see these oscillations very regularly in humans as well as in animals. And if you take a problem, look, there are at least three axes. Let me just say it this way. You have to know the drug class, how much did you give, and then the age of the patient. There are at least three axes. So depth of anesthesia folds them arbitrarily into one axis and just assumes that all the drugs work, everything works the same as long as you get the same number. That's what's happening here, all right? And I think that it's very easy to see once you look, just plotting the data. I didn't go over any mechanisms at all. We could have a long discussion about that. Just plotting the data just shows you that. And you know who's losing out? The patients are losing out. Because anesthesiologists don't track their brains very well. So what are you going to do? If you want to make sure that someone's anesthetized, you're just going to overdose them. Right? Because nobody wants to hear this, oh, I was awake while they were operating on me. So you can just infer right off the bat the patient's getting overdosed, and they certainly are. But yet this is, and again, to put it into further context, this is the strongest EEG signal there is. It's stronger than what you see in sleep. It's stronger than what you see in, let's say, cognitive studies, when looking at patients with epilepsy or these sorts of things, because there's no noise once the person becomes anesthetized. You're seeing dynamics coming directly from the brain. We have the strongest EEG signal around, and we don't use it as anesthesiologists. And as I said, the patients are losing out. So, again, I needed a counter story for this. So last Friday, I clicked my heels 3 times, and this email came, all right? It was from a CEO of an AI startup, and here's what he said to me. It says, using real-time data, we aim to develop an AI-based system able to predict real-time hemodynamic risks and present explanatory explanations of factors, contributions to that risk during general anesthesia. The informed risk prediction enables anesthesiologists to plan an appropriate course of action to avoid such risks and take best possible decisions for their patients, right? And he says, to do this, we need some big anesthesia data. All right, so I folded all our data up and I sent it to him right then. Yeah, right, exactly. This happens a lot, and this isn't the first such email that I've received, and I'm sure there are those of you who received similar emails. I mean, what we see is data is not information, all right? And so as I was thinking about what to extract out of this and sort of keeping the science in data science, so don't let the ease of large-scale data collection and analyses change or overly simplify scientific questions to be addressed, all right? I think for those of us who are statisticians, it's incumbent upon us to help make sure that inference paradigms remain in place and don't get thrown aside just because we can compute things automatically. And I like to talk about the Box-Tukey paradigm. This is the way I learned to think about sort of scientific inference. The whole idea of exploratory and confirmatory analysis coming from John Tukey, and then Box's idea of iterative model building. And the same thing applies, I think, in where we are now with our data sciences, and I think this is something which is really can't be emphasized enough. Training and education are going to be key. And I'm thinking about like experimental design for observational studies, because if you're thinking about it, a lot of putting together a data set or data sets is really, you're saying you have observational studies. Cross-training is really, really important as well, because the areas of expertise, training in some area of expertise along with some area of data science. And then retraining like I did. I mean, I retrained. I had to learn. There was stuff out there I needed to learn that I didn't learn in graduate school, and I think that's going to be a continuing issue sort of going forward. And cross-disciplinary collaborations are essential because we have to think about who is really intellectually responsible for the findings that we report in studies. So I'll stop there. Thank you.
Emery, maybe we can keep your last slide because I'll say amen to this.
Okay, all right.
We can invite questions from the audience. Or maybe while the audience is thinking, maybe I have a question.
Sure.
Do you know of cases where misuse of this has led to dramatic consequences?
I mean, I have to scratch my head to think of like dramatic consequences. But I think the fact that, so just to put it into perspective, we have 60,000 anesthetics a day, 60,000 surgeries in the United States. So there are like 60,000 surgeries per day in the United States. And at best, let's say 25% of those, there's some sort of EEG being used. And a high fraction is probably with BIS because they have the largest market share, right? So we don't have to have like sort of these very like sort of egregious faults. There's just a huge fault built into the system that happens probably to at least 80% of the patients, or if not more, who are having surgery, and that just shouldn't be. So I think it's still quite an issue. A question here.
So trying to get at the big anesthesia data.
I think we're online, so I think if you can use a microphone.
Trying to get the big anesthesia data that the company is looking for. So you say that there's so many surgeries done a day and under anesthesia, of course, and some of them use EKG. Can we collect that data? It's clearly not an experimental setup, but can be used in some ways?
Right. No. So two things. It's easy to collect. We have electronic medical records now. But it should be done prospectively, right? Don't come to me and ask me for my data, without some, and I don't know if you've ever talked to an anesthesiologist before you've written me. I have no idea what the person understands scientifically. And I see a lot of this where the people write and say, oh, I've got a new classifier. Can I try it on your anesthesia data? I mean, that's what I'm talking about. In other words, the science is not there. And I think that's what we have to put the emphasis on going forward.
Thank you.
Brian.
Can I just speak loud? No.
Thanks. That was really very interesting. Thank you, Emery.
Thank you.
I wanted to ask you, there are two things that came to mind. One is, first off, I don't understand Medtronics in this story, right? You'd think that they would want to make their algorithms better. So I find that the situation there a little bit puzzling. And in fact, to simply predict the BIS, given that you have a lot of data, one could imagine that you might be able to build a lookup table form to go and sort of deconstruct it.
Probably could.
Okay. And then the other question is, in thinking about mechanism, these measurements are kind of brain-wide measurements.
Right.
And I'm wondering if there's any spatial localization to the EEG contrast measurements that are also insightful.
Right. So just to respond sort of seriously to the first question, yeah, and in the spirit of full disclosure, we're working on better algorithms which could be used commercially right now, just to be frank. And as far as the second one is concerned, yes, my colleague Patrick Purdon has done a lot of work on source localization for like the oscillations. And he has a couple papers which came out a couple of years ago, and also one which will be coming out in a few months or so, basically giving us some idea of where the oscillations are coming from. And taking advantage of this data like we talked about last night, where you have patients that have electrodes implanted in their brains for epilepsy surgery, and you can get them to do a controlled anesthesia study on them to sort of learn about where the drugs are acting. So we actually know a lot, and we've also done the same thing like in non-human primates. So this is, to have an algorithm like this sort of dominate the market, and then people going, I'm not going to use it. It's just a travesty. I can't say it any other way. And I honestly believe it set our field back, because the things that I'm showing, just those plots, they should be in the head of every anesthesiologist essentially in the country and thinking about taking care of their patients, particularly the ones for kids. I mean, that's inexcusable.
Thank you for your talk. In the middle, you made a comment that when you have had a successful data-driven study, you've tried to squeeze out most of the methods from it in different studies. Do you have any practical tips of how we can achieve that?
No, that's a very good question. And what I found was, so I'll explain what happened in this particular case, and maybe we can extrapolate from that. So what I noticed in the neuroscientists doing was they would spend all this time to get spikes, so ones and zeros. And then the first thing they do is they would smooth it, right? I'm like, dude, what, you spent all that time collecting it, why you just threw it away, right? And so I said, how can I just continue to keep the ones and zeros and do a regression? Or how can I keep the ones and zeros and run a filter? So that was point processes, and then just basically the GLM with a essentially logistic model to first order. And this had been known. I mean, if you go back and look at this paper in 1988 by, I'm forgetting his name, at Berkeley. I'll think of it in just a second. Brillinger, right, exactly, Brillinger, thanks. Brillinger had said do this. He said, he actually wrote this out in one line, and nobody else paid any attention to it. So, and then what I realized was there are a lot of other situations where we have zeros and ones in neuroscience. So behavioral studies, did the animal get it right or wrong? That sort of thing. So that's what I'm talking about. So essentially we had a paradigm, and then Statespace was very important because there's continuity in a lot of these things which is not taken into account. So that's what I'm referring to. And so there's a whole series of things that we did after that took advantage of that sort of, because the problems had the same model structure, essentially.
So can I ask you again about your last slide? You had this plug to education, and what does it mean to educate people in data science? Is something that we are struggling with. Is it statistics? It's computer science? It's everything. So could you expand? What are your thoughts about education in data science?
I think that's a good. So again, let me explain it by example. So I was on the NIH panel for the Brain Initiative. And one of the things we talked about was educating the next generation. In other words, the people who were doing quantitative work had to learn the neurophys techniques, understand how they worked, what the properties of the data coming out of that were. And it wasn't sufficient enough for just the neurophysiologists just to be there and just start using methods and not understanding them. So we have to create courses. That's why I gave the Woods Hole example. That was I was in that class. I was junior faculty. Everybody else was either a graduate student or a postdoc. It was just like, the 10-year-old in first grade, essentially, But we need to have, and with web possibilities now, these sorts of things are much easier to set up. But we can't produce these very complex algorithms, complex from a statistical standpoint, complex from a computational standpoint, but they're easy to use because all you have to do is feed your data in, and nobody understands what's going on, because that's going to kill us. That's what I had in mind.
Steve.
Yeah, I wanted, let's see if this works. Does that work? Yeah, whoa, sorry. So I wanted to expand on that. The experimental design for observational studies is music to my ears because I'm an epidemiologist. The whole field is based on that.
Right.
And all of our graduate students take data science courses. Almost no data science students take our courses. And the question is, how does this not, and there are other courses given in statistics in this,
Right.
but it doesn't seem to be absorbed into the culture of data science, particularly coming from the computer science realm, that this is necessary. And so how does this, aside from just offering courses, the courses are there. And when I've discussed this, I'm often told, well, there isn't room in the curriculum. They have to learn these higher level techniques.
No,
so how does this become part of the culture of data science training to make what you have as number one on your list, in fact, a number one priority that might be taken in the first year?
I think we have to, in other words, we have to make it part of the required curriculum. I just don't see any way around it. And I see you have, I think Guido Ibbons is speaking later on today. I'm sure he'll have some comments on this also. But it just seems like to me, these people, when someone calls and asks me for my data set, right, they want to do an observational study. Basically. That's what they're telling me. And I would say, do you have your observational study certification? That's what I'd like to ask. Are you certified to do this, And it's interesting because we set up a statistics institute at MIT about 6 years ago. And we started pulling together all the courses. We had a lot of probability courses, statistical inference, data analysis courses, all. I said, is there an experimental design class? And we finally found one in AeroAstro, right? Because, they have to do very well-designed experiments. It makes total sense. But I think we have to bring this out to the fore. And that's why I wrote it as number one. Because when people call me up and ask me for my data, they want to do an observational study. And I want to know, are they certified to do it? Russ?
So this has been a really fun discussion, and thanks for these comments. So you've lived through at least a couple of hype cycles around particular, types of methods. And I'm curious how you think about, for people in the audience who are thinking about, how do I think about the latest, greatest method, and what should I take from it versus being kind of skeptical about, the hype? I'm wondering how you think about that.
Well, I think you have to always have a healthy degree of skepticism. I think there's no way around it. But where I'm able to really get my head around it, at least conceptually, maybe not technically, but at least conceptually, is when they tell me what they want to do with my data. And they say, well, I want to study hemodynamics, and hemodynamics were in the operating room or hemodynamics in the ICU. And do you realize the hemodynamics are tightly linked to whatever anesthetics I give, right? And so that's the sort of thing that where with just a few questions you can usually debunk, whether or not someone truly understands what they're doing, or do they have a plan for a well-designed study, which is what I think. I just can't see how we can teach data science without the experimental design course being like a fundamental part of the program, because essentially they're observational studies most of the time. That's what we're saying.
Other questions?
One more here. One there.
Thank you for this talk. And I found it particularly interesting because I woke up in the middle of a sinus surgery about 8 years ago. And I actually found it to be not an unpleasant experience, surprisingly. I guess my question is, how do you set so you have a decision problem here in terms of whether you want to give me more or less.
Mm-hmm.
And you know, how should we think about setting this problem up? What is the goal of a better BIS in terms of what outcome are you trying to maximize?
That's a good question. So let me just say the effects of the drugs, like the drugs we're talking about, like propofol or the ethers, right? They do look pretty much like this, okay? And I find it hard to believe that you woke up and your EEG wouldn't have shown some sort of indication of it. It's now next to impossible. And so what I would do is, you see the first large set of, see where it says alpha, slow alpha oscillations? I would dial you down to just that stage, because that's the stage where you're sufficiently unconscious that, you know, I don't have to worry about you waking up, but you're not overdosed, basically. So that's what I would shoot for. Now I can see that. I can see that in the panel there, and you see how the oscillation has high frequency oscillations and then it has low frequency oscillations. As we get older, we lose the high frequency oscillation. We'll have just the low frequency oscillations. So I'd put that in the context of how old you are and what I'd expect to see. And we've looked at enough patients to have a really very, very good sense of that. I want to teach the anesthesiologist how to read this and not rely on, I mean, to think that you could simplify this problem down to something which is just a number between 0 and 100 is an embarrassment, quite frankly. But however, understanding these patterns, in other words, I don't want you to just recognize the pattern. I want you to understand why that pattern comes about. How is it being generated by the neural circuits? Then when an anesthesiologist is taking care of you, he or she is really using neuroscience to take care of you as opposed to pharmacology. That's what I would recommend.
Earlier you said that this pattern may depend on your age also.
Mm-hmm.
So the anesthesiologist will have like a wide array of things that they need to recognize depending on your age.
But they're very stereotypical, that's the thing. It's not a large set. In other words, it's not one dimension. It's 3, maybe 4 dimensions. But it's very, very tractable.
One last question.
Yeah, thank you very much for the talk. It's a refreshing perspective that you bring. I'm curious on what types of arguments do you bring to the fore when you've got like your Vanderpool example, you're getting 75% of the prediction value out, but the deep learning model comes along and gets 98% out. Presumably the Vanderpool model gives a better explanatory example that you can use to infer the underlying processes.
No, that's a very, very good question. And I would go with the Vanderpool because I understand how I construct it. I understand, what it means. And then, as sort of simplistic as this sounds, I would look in the residuals. What did I not explain? What did I miss? In other words, and then I think it is a fair question to say, is there something that the neural network model is telling me about the physiology that I missed? Because that's what I'm interested in. I'm not just interested in their fitting just for fitting's sake. I really want to make a scientific inference about the properties of the system that I'm looking at. So if they came out and sort of beat me, that's fine. That's great. But nevertheless, then you have to tell me what new science you figured out at the same time. Was there some sort of new pathway that you detected that we weren't aware of that we should now go and study? I need to know what the additional inference is, basically.
All right. We want to thank you, Emery, for a wonderful talk.